Hey, it's Peter here with my FSHD. All right, we're back. I know you're groaning. I can hear the groans from across the pond. Another podcast on FSHD gene therapy. But I know a lot of you are really busy and some of you have really short attention spans. You're interested in the big picture though, probably. Maybe not so much in the really nitty-gritty details of the science. Some of you are, which is why we put the long podcasts out there. I think it's important for people to have the opportunity to understand why we have our opinions of the science and how we interpret the data to form those opinions. It's not random. There are points of contention, and some of those have been brought up to my attention. And so in this new, we're calling it an all-in-one podcast, we're going to try to put everything all together, hitting the high points. But there will be some new stuff because we have had some questions that have been raised. I always love that. I always love when people comment. You don't have to tell me it's great. You're more than welcome to ask questions and say, this doesn't seem quite right. Because we want to get the right answer, right? Even if the truth gets you arrested in the UK or shot in the US. And, well, again, I understand they couldn't find the truth if it was covered in maple syrup in Canada. But we want to get the right answer, okay? And if we can. Sometimes the answer is we don't know, we just got to do the experiment. Actually, that's kind of what a clinical trial is. Well, it's not even kind of, that is what a clinical trial is, testing a hypothesis. What we, though, want to get to is, do you have the foundational data? Have you done enough ahead of time to warrant testing the hypothesis in FSHD patients? That's really the question. And let's get to it. I'll do my best to keep it as short as I can. So as I mentioned, if you don't already know, there's already 5 podcasts out there, 2 covering kind of general gene therapy for FSHD, these up here. And then our EPI-CRISPR podcast is kind of broken into 3 parts. And yes, we know it's actually quite long. It might be about 4 hours total. But this one is going to be the all-in-one, hitting the high points of all of these podcasts up above. Try to get it, just kind of boil it down to something that's easier to listen to, easier to watch, and easier to follow. But again, if you're really interested in the nitty-gritty and really getting into the data and the concepts behind what we're talking about, I think they're pretty good. We do our best. Of course, we always come up a little short, but that's okay. And then, as always, feel free to prove me wrong. Okay, I think that's a good slogan to have these days, is when we want to have a civil discourse about the topics that may be controversial. All right. And as many of you know, but many of you may not know, since we're doing video now, everything is available on our YouTube channel. You can subscribe to the MyFSHD YouTube channel, or just, you don't have to subscribe, but you can just, I'm not actually sure what subscribing does for you. But you get in there and you'll be available to see all the videos. And these are the 5 videos that I was talking about. And this one will be posted up there as well. In fact, it should be as soon as this is done. All right, I got to have some disclaimers, of course. Myself, Takako and Crease, are founders and have equity in Renogenics Inc., an FSHD company with a CRISPR inhibition gene therapy program. So if you think we have a conflict of interest, if you think we're just bitter or angry or worried about competition, that's fine. You can have your own opinions. We're not hiding the fact that we do have that conflict of interest, but we do our best to be unbiased. We are investigators in a research lab focused on FSHD at the University of Nevada, Reno. But that being said, the MyFSHD organization, MyFSHD podcast are not associated with the University of Nevada, Reno, or Renogenics, or any other foundation or company or individuals. The purpose of the podcast is for educating the FSHD patient community about the science and the scientific process as it relates to the FSHD field. Man, a little bit about the scientists, that we're just like you. Maybe that's not so comforting. You wish we were better. And many people don't know this, but we do not raise money. We don't even accept donations, right? We just do this because we want to, okay? The opinions we don't get paid, trust me. Do this on our own time as well. The opinions, interpretations, and comments presented here are those of the speakers and not endorsed by the University of Nevada, Reno, Renogenics, or any other entity or individuals. All the data is publicly available. We don't do anything that's confidential. We just, there's some data we're going to miss. There's some data we can't include. So we, if there's other data we're not aware of, you feel free to make us aware of it. But just sometimes we ask for some things that maybe are out there and we're just not aware. And importantly, this presentation does not represent medical advice and should not be used to make medical decisions. And if you're one of those people that has one of those aforementioned attention deficits like I do, I'll just give you my executive summary of my opinions of the data right here. It kind of covers everything. Okay, there's kind of 5 points overall, and we're going to go into these a little bit more. I'm concerned about the EPI-321's use of AAV-RH74 as a capsid. Okay, it's well known that the viral doses for this capsid need to be extremely high and are borderline safe and have actually led to some deaths. There are much better AAV capsids now available for use in FSHD gene therapy. Other companies are using them. They could do this if they want to. The clinical trial dose is very low, so it's likely safe. But it's also, in my opinion, very unlikely to show any benefit whatsoever for patients. In fact, I believe that you're unlikely to even learn anything about EPI-321 in these human experiments, and that ultimately, if they choose to stay in the FSHD space, they're going to have to repeat everything again with new doses and likely a new AAV capsid. Okay, so what's the point of doing the trial? That's my if you want to take it home, that's my opinion. Secondly, I have concerns about gene regulation not actually showing muscle-specific expression, despite using a well-known muscle-specific promoter called CK8E. Okay, this is actually the promoter used in Duchenne muscular dystrophy trials, and it's portrayed as muscle-specific. But when you look at the data, and this was a point that was brought up to us, because people are like, how can you say it's not muscle-specific? It's CK8, which is a muscle-specific promoter. We look at the data. Okay, a lot of people read titles of papers, a lot of people read abstracts, listen to what the authors and people are telling you or spinning for you. We look at data, and we'll go through that. This is some of the new data we're going to go through. Okay, CK8E promoter is used in the DMD gene therapy trials, okay, and EPI-321 does say in one of their abstracts that they are using this as their muscle-specific promoter. It should be muscle-specific, but everything is context-dependent in the case of the entire transgene when you make something, and we've learned this many times when we make animal models. This is important to some degree for safety. I'm not really worried about the safety in this case, but I'm more worried about it because of durability. When you start expressing gene therapy cargoes in tissues outside of skeletal muscle, you get more exposure to the immune system and leading to more chance for durability issues, which quite honestly they see, in my opinion. The preclinical data shows that, as I mentioned, it does not have the gene expression, tissue expression profile that one would expect from CK8E. That's part of it, but it actually looks like it's expressed in tissues that it shouldn't be expressed. And combined with IAVRH74, which infects many, many tissues just equally well or even better than muscle, that targeting and the wider gene expression activity raises concerns over EPI-321 transgene expression. Again, it's not safety concerns per se, although myocarditis, as we'll talk about, could be an issue. It's more an issue of activating the immune system and leading to durability concerns, meaning that the cells that you so-called fix or cure get eliminated from the body. We have concerns about the preclinical data not testing efficacy in FSHD patient target tissue, which is differentiated skeletal muscle. Most of the data was collected under idealized culture conditions not relevant to FSHD patient muscles. It's a disagreement in how we do the experiments. Obviously we have a different opinion than they do in this respect. Concerns about the durability of the cargo expression in patient muscle cells. Large mammal data, meaning dogs really, but also in pigs, shows that the immune system clears cells expressing SP and SA Cas9 proteins, okay? The preclinical non-human primate data from EPI-321 strongly suggests to me, and agrees, that this is happening with EPI-321. Okay, their data is too short, and they are either ignoring or not addressing key concerns about durability. And overall, number 5, I believe they just are prematurely going to clinical trial, likely to have no benefit to patients or scientists. I don't think much is going to be learned from this. This is my opinion when I put it all together, okay? And why are they doing that? Well, I have opinions on that too, but I'm probably better off keeping those to myself. All right, so getting through things briefly, just want to make sure it's very clear. It's very important in science to give credit where credit is due. We did not discover CRISPR or CRISPR inhibition. Okay, CRISPR repeats were first identified in 1987. CRISPR really came of age in 2012 with Jennifer Doudna and Emmanuel Charpentier when they actually put the CRISPR system together and showed that it was a great a tool for genome editing. George Church and Feng Zhang then showed that it could be editing in human cells. Okay, and then importantly, dead Cas9 was actually developed by Stanley Shi as one of the inventors of that. And he actually is one of the founders of EPI-CRISPR biotechnology. But he actually did invent and develop dCas9 for using CRISPR as a genome targeting system to bring activity such as repression or activation to a point in the genome. Okay, so our role in developing CRISPR was using this dCas9 repression system for the first time on the human repeat genome for the D4Z4s and showed that it's viable for FSHD. Okay, that's where we come in, and that's why it creates the CRISPR goddess. They probably, I'm sure back here, had never even heard of FSHD, and we were already showed that it was viable for FSHD. Okay, CRISPR is essentially genome targeting. Most of the time you hear about genome editing. Okay, that's where you cut the DNA and change the sequence. You can actually, in the lab, insert sequences, fix the sequences, a lot of things you can do. But for FSHD, we're really talking about dead Cas9, which is dCas9. And this is where you recruit an activity. Instead of the CRISPR machine is able to very specifically find a site in the genome and then bring in any sort of fused effector domain. So you can turn genes on, turn genes off, make them glow. You can do all sorts of different things. And this is a wonderful scientific tool for biological research, biotech, and disease treatment. Okay, different ones have different aspects. All right. So those of you who have talked to me on Zoom calls, which is quite a lot of you, I've done several of them, a number of them this week. So I had my friends down in South Africa that I met this week. I did a number of South Africa calls. I would like to talk about FSHD in comparison to DMD. To show an example of the difference between healthy muscle, DMD muscle, and FSHD muscle, if you're looking online on the YouTube channel, the video, in DMD muscle, all the cells essentially are lacking dystrophin. They're all missing something, a key component that they need. Part of it is for structure integrity. It also provides a key signaling functionality, and that's important in muscle health. Okay, in FSHD, it's a little bit different. In FSHD, what you have is your cells are genetically the same, but they have the potential to express DUX4. They don't actually all express DUX4. And we're going to show you next slide that they don't even all have the potential to express DUX4, but let's just start with baby steps. You have the cells in FSHD muscle are poised to express DUX4. And if you're looking online, a few of them do, a very small number of them do. And then over time, another one might pop and due to stresses or immune responses or oxidative stress or who knows, hormones. And another one expresses DUX4. And over time, if enough of them express DUX4, the muscle can actually start to really go down a pathological cascade, and you end up getting muscle weakness, and you could even lose a muscle. All right. But as I mentioned, it's a little bit more complicated than that in FSHD, right? So it's dominant gain of function. Duchenne was loss of function, okay? But again, in Duchenne, where all the muscle cells have the same defect, in FSHD, depending on your epigenetic states of your cells, some have the potential to express DUX4, while others do not have really much potential to, they're resistant to expressing DUX4. And this is where your epigenetics come into play. You may be genetically the same at the locus throughout all the cells in your body, but epigenetically, you are a mixture. And if you've taken our research test, where we look at the methylation, you actually get to see that. You see ranges of methylation, okay? Every cell has a slightly different methylation pattern and different level of methylation. And the higher the methylation, the less likely it is to have the capacity to suddenly express DUX4, and the lower the methylation, the more likely it is. And so what we give you is an average of your methylation on your FSHD contracted chromosome. But, if it's very low, you like this one here on the left, you have, most of your cells in light green have the capacity to express DUX4, but there's still some cells here in gray that are they're methylated enough that they're never going to express DUX4. And then you have cells in pink that are actually expressing DUX4. If you have slightly higher numbers, maybe in the teens, approaching the 20s, for your quartile calculation, more of the cells are gray, meaning that you actually have more cells that have a higher methylation, a healthy level of methylation, and refractory epigenetics. You still have a large number of cells that have the capacity to express DUX4, but it's changing, right? And then when you get up into the mildly affected or the asymptomatic stage, it gets even further down that path where even more cells are refractory epigenetically to expressing DUX4 and less likely to ever express it and then lead to pathology. So it becomes a kind of a statistics and chance game. The lower your methylation, the more likely, the more cells you have that have the capacity to express DUX4 epigenetically, and the higher your methylation, the more you're epigenetically refractory to expressing DUX4. And this kind of correlates with your disease severity and the variability that we see in families. So now if you're going to come in here with a therapeutic, and if you think about this, if you're looking at the video, come in with this dark green therapeutic, like a low dose AAV, that's only going to hit, say, 10% of the cells or 20% of the cells, well, that's going to actually matter because one, some of the times it's going to hit cells that are refractory already to FSHD if you're a mild case, and so you haven't really done anything. But you also are going to shift some of those cells that have the capacity to express DUX4 further to the healthy state. Okay, so a low dose therapeutic might actually, push you even further into a milder asymptomatic. Now, if you have very low methylation, where most of your cells have the capacity to express DUX4, well, one of these gene therapies might not do a whole lot for you because most of your cells are going to still have the capacity to express DUX4. Okay, but conceptually, you might think, well, maybe it dials it back to a less severe case. Now, we're talking all this time about DNA methylation, but I think this actually is, again, too simple. Because really what we're talking about is not just increasing methylation in order to get epigenetic repression, but you have to change the entire epigenetic state of the locus, okay? So if you're looking online, you need to understand the difference between the FSHD epigenetics at the D4Z4 and the non-FSHD epigenetics of the D4Z4 is not just DNA methylation, okay? Which is this little triangle here. It's the DNA methylation is associated in the healthy case with a huge number of repressive proteins and factors. First off, the histones have histone tails. Histones are the proteins that the DNA is wrapped around and is compacted very tightly, and they have repressive post-translational modifications covering these histones, and which recruit in more histone-modifying proteins, heterochromatin protein 1s, cohesions, chromatin remodelers, SMCHD1, NERD, HDAGs, histone deacetylases, polycomb factors, small RNAs that keep the silencing going and help maintain through generations, and DNA methyltransferases, okay? So you have this entire heterochromatic complex that is assembled at the healthy locus, okay? And DNA methylation is one of the epigenetic components. It's the easiest one for us to assay. So from a diagnostic standpoint, the difference is methylation. From a biological standpoint, it's actually very different because now if you go over here and you look at the FSHD side, where you've lost the methylation, it's not just a matter of not having methylation. You actually have, again, the histones that the DNA is wrapped around all have chromatic or active or potentially active histone tail modifications, which recruit in their own chromatin remodeling factors to keep it open, transcription activation complexes, histone acetyltransferases, ATP-dependent chromatin remodelers like imitation switch and SMARCA and BAS1, CTCF, which is it's involved in 3D organization of the nucleus, where you end up with domains of active and domains of inactive parts of the genome, and all these sorts of things. And so it just adding some methylation to the FSHD region, the question is that going to actually do anything, right? So the difference, now the thing to remember is I just showed you, we talk about the difference between being symptomatic and asymptomatic is not average levels of methylation, even though that's the readout that you get in your reports. The difference is actually the number of cells that you have that are in either this right side potential to express DUX4 or this left side unlikely to express DUX4. And the milder you are, the more asymptomatic you are, the greater the category on the left, and the number of cells. It's a numbers game, and the number of cells that have the capacity to express DUX4, and the greater the number of cells, the earlier onset and more severe you are, is the trend, okay? So, if you're thinking therapeutically and you want to change this epigenetic switch and go from an FSHD state to a healthy state, you can't just add methylation. You have to change the entire epigenetic components associated with the methylation. And the time when you do this actually is during DNA replication. Unfortunately, FSHD differentiated muscle, which is your target tissue that's going bad, is not replicating, okay? But we'll get to that in a sec. Just keep in mind, methylation is the readout, okay? And it is involved in establishing repressive chromatin after replication. But just methylation itself, that's just sitting there, just changing the methylation state on a piece of DNA in a cell that's in G0, might not do anything, okay? All right, so let's talk about CRISPR inhibition. Okay, you guys know all about this, right? There's two kind of ways that you can do CRISPR inhibition. One is recruiting a repressor protein where you're essentially going to drop a big off switch. And the other way is to modify the epigenetics, bring in a modifier. You can bring in a DNA methyltransferase. And this is what EpiCRISPR is doing. So essentially the idea is in FSHD1, right, you have this contracted chromosome 4 that is epigenetically on. The non-contracted chromosome is epigenetically off, and your chromosome 10s are also epigenetically off. And so the trick is you want to change the activity state of the contracted FSHD chromosome 4, which is where DUX4 is being expressed from. Okay, so if you're going to do it with transcription repression, the idea is you use dead Cas and the targeting capacity of the dead Cas to bring in a transcription repressor and essentially drop an off switch on the active DUX4 gene, and all the other DUX4 copies are already off, but you're still going to drop another off, and this is targeted transcriptional repression, right? The other way to do it is similar, but it's with DNA methyltransferase, right? Bring in a DNMT, DNA methyltransferase, and methylate the region. And my animation didn't exactly work, but that's okay. And it's going to be very similar, except that this putatively is going to modify the DNA with methylation, okay, and then turn it off. Now, in the previous case, you're actively shutting things off. It's not clear that adding DNA methylation in FSHD tissue where the cells are not replicating is actually going to shut it off. And that's why it's important how you do your preclinical data to actually make sure you're testing the right activity, okay? So I actually have a question mark on whether in FSHD patients, this would actually lead to being off. But conceptually, the reason one might like this better than the targeted repression is if you ever somehow lose the chromatin modifier decast fusion, either a transgene silencing event or something, or who knows, or stability or whatever, the epigenetic state may be maintained, in which case then you maintain the off, where in the other case you might lose the off. Now, the issue, this is a bigger deal if you have replicating cells. Again, in FSHD, the target tissue is not replicating cells, okay? Now, there's one other thing that kind of is like a way to do this that has been talked about. There's a group in Japan that published a paper called Hit and Run, okay, and we'll talk about that in a sec. And what they showed is that DNA methyltransferase actually needs the transcription repression fused to it in order to actually work in FSHD. And again, they were working in replicating FSHD cells. So this is the other way to do it, is to combine the two approaches, right? If you can fit it all in. And, you know, it's possible to fit it all into AAV now these days, potentially. But the idea would be that you have DNA methylation and a transcriptional repressor. And so then it doesn't really matter, right? This was actually going to work on non-replicating cells. And really, though, the methylation may or may not matter. The key is going to be you have targeted transcription repression, okay? And honestly, I suspect this might actually be what's going on, although I don't really know. I hope it actually is what's going on, because this will work better than DNA methyltransferase alone. All right, so again, Remember, it's not just about increasing DNA methylation. You got to change the entire epigenetic state of the locus, all right? And so if you're going to go from right to left, go from FSHD to a healthy state, it's really going to help if you have the targeted transcription repression and the DNA methylation. But if you want to check the epigenetic state, you're also likely going to need replication, okay? And that's really the fundamental question. If it is just about DNA methylation, can you really accomplish this change just by adding a little methylation in non-replicating cells? And I don't believe you can. In fact, I'm not aware of any data that anybody has ever showed that says you can actually even methylate non-replicating cells, much less change the epigenetic state, the entire state of a locus in non-replicating cells, right? So that's the question. Is this really flipping an epigenetic switch? Or are you actually just shutting it off? Now, therapeutically, it may not matter because shutting it off, but scientifically and conceptually and how you might assay the therapeutic, it does matter. All right, so gene therapies for FSHD are going to be delivered systemically in vivo using a virus, and most typically it's going to be adeno-associated virus, or AAV. And the thing to know about these AAV, since it's going to be injected intravenously in a single one-time injection, first off, everything has to fit into a single virus. But secondly, no matter how good your virus is, it's going to go not just to skeletal muscle, but to a lot of other tissues, okay? Nothing's actually perfect, but there's different levels of effectiveness. So you'll see phrases like effectively transducing or effectively infecting skeletal muscle. But they also effectively, for example, the one that EPI-CRISPR is using very effectively transduces liver and the heart and the kidney and many other tissues. Okay, so it's not just about getting into skeletal muscle. You actually want to avoid getting into these other tissues as much as possible. And there's a number of reasons for that. Okay, as we mentioned, FSHD gene therapy, you have to get everything into a single viral capsid. We talked about this a lot in the earlier podcast, and that's kind of a trick. Everything needs to fit. But once you get it in, we've developed as a field a lot of tools, 2D and 3D muscle cells, the FlexTux4-like mouse model that my lab developed, the humanized xenograft mouse model for FSHD. There's several of them out there. We now have an FSHD-like pig model that you can do, and then healthy non-human primates. And the key is that once you get everything into this virus, because you have to change things up, you have to pack everything in, and the devil is in the details. And just because you put something together that conceptually might work really well, or even works really well in cell culture, the question is it doing what it's supposed to do? Are all the components functioning correctly in vivo in the system? And yeah, sure, I guess you can try it out in a patient, but you know what? A lot of this can be checked out in a mouse model for FSHD, pig model for FSHD, or a non-human primate, because there's a lot of things that can go wrong that you could find out before going to a patient that would actually save patients from undergoing a trial that was doomed to fail from the start if the preclinical data had been done correctly. All right, so I mentioned before, there are 3 companies that are publicly out there. There may be more, but these are the 3 that have publicly come forward for gene therapy for FSHD using CRISPR inhibition. EPI-CRISPR, of course, they're in the clinic. Okay. Now, the key is they're all very different. I guarantee they're going to be different because there are different dCas9 proteins you can use. There's different repressors you can use. There's different guide RNAs you can use, different promoters, different capsids. You know, you could have 20 companies doing CRISPR inhibition for FSHD, and the devil's in the details, and they all can be different. The one thing that we do know about EPI-CRISPR is they say they're doing the de novo DNA methyltransferases. Possibly, I suspect they might have the crab protein for repression. I don't know. But they're definitely using AAVrh74, okay? And that's the one we're going to focus on today, right? They do have their first in-human study of EPI-321 and FSHD, locations in Atlanta, Sydney, and New Zealand. Phase 1/2 open-label dose escalation study to evaluate the safety, tolerability, and biological activity of EPI-321, an Rh74 delivered epigenetic editing therapy in adult cells, right? So they're really doubling down on the epigenetic aspect of this, claiming that they are flipping the epigenetic switch from an FSHD state to a healthy state. Okay. Again, it may not matter if that's really what they're doing or not, but I will question whether they are actually doing that. So here I'm just going to present my scientific interpretations of the publicly available data from EPI-CRISPR Biotech, okay? Integrate their data with what's known in the NMD gene therapy field, including preclinical data for other gene therapies, prior and current NMD clinical trials, and form my opinions and potential impact on the presented trial design on FSHD patients. That's what I've been asked to do by you all, so that's what I'm going to do. All right. So EPI-CRISPR, just briefly, you can look them up at epicrispr.com. They're a platform company. FSHD is one of many diseases they work on. I actually like the company a lot. I like the platform. Their idea is that they have these specialized dCas9 proteins that are really small, so you can fit in a lot of additional cargo and regulate genes in different ways. Okay, platform technology, and their approach in FSHD is to silence the DUX4 gene. Okay, this is what EPI-321 looks like. They have their dCas Onyx, which is their specialized super compact dCas protein. Their modulator, which is a DNA methyltransferase. They have what they say is a muscle-specific promoter, which is CK8E, which quite honestly doesn't appear to work as well as it should as a muscle-specific promoter. And then you have your guide RNA. Right, so this is going to be the repressor. The guide is going to tell the repressor where to go, and the muscle-specific promoter is going to tell you which cells to be active in. Okay, that's the idea. It's all going to be delivered in AAV R874. All right, so let's go to the videotape. All right, so this is from one of the posters that they've presented, and what they show is they're actually going to go into 6 different cell lines of primary FSHD myoblasts, and show that if they put in EPI-321, they can get about, on average, I'm guessing looking at this, at about 12 viral genomes per cell. And if they get about 12 viral genomes per cell, they're going to get about 40% repression of DUX4, okay? In vitro in idealized conditions. Now, we don't know exactly how they're doing the experiment, but if you follow exactly what they say, sounds like they're infecting replicating cells and then driving differentiation, which is kind of what's shown over here for their apoptosis assay. And so if that's actually true, then as opposed to treating cells that are already differentiated, then they are actually using DNA replication to their advantage to help establish repression that might not otherwise be established. That's a concern of ours. But regardless, now they're going to look at apoptosis. Apoptosis, if you're not familiar with the term, is programmed cell death. And if you overexpress DUX4 in just about any cell, the cells will die. They hate expressing DUX4. Now, primary muscle cells from FSHD patients in our hands typically don't have a lot of apoptosis. Oddly enough, these every green dot here is a cell. This is an assay that shows you by fluorescently labeled cells that are undergoing apoptosis. There's fragmentation of the DNA, activation of enzymes that cause cell death, and they're able to detect that with this special kind of assay. And so the more green you have, the more cell death you have. And you can see here that they are reducing cell death, it looks like to me, by about 30% in FSHD. Honestly, I look at this and I'm concerned about how they grow their cells because we just never see anywhere near this level of apoptosis, but whatever. All right, so then of course they're all about epigenetics. So we're going to go to DNA methylation, and now they're going to infect cells, and they're going to look at the DNA methylation with EPI-321. And honestly, I've never seen DNA methylation shown this way. The untreated cells have pretty high methylation, and then honestly I can't interpret this, but if we'll take their word for it, they increase DNA methylation by 8.5% somewhere. I don't know if it's just one little fragment that gets increased. Honestly, I got no idea what this means, but we'll take them for their word. They're increasing DNA methylation by 8.5%. Interestingly, in this now suddenly they get like 99% reduction of DUX4 in this assay. Not sure why that is. We've never seen reduction of DUX4 down that far in our hands. However, DUX4 target genes only go down about around 25%. Remember, DUX4 is a transcriptional activator protein and activates a number of genes, represses some genes. So there's a DUX4 signature in FSHD muscle that you can follow that tells you DUX4, which is really hard to detect, was expressed. And if you look for the reduction in the DUX4 signature, that suggests that DUX4 is going away. So you have a 25% reduction in DUX4 signature and maybe a 60% reduction in apoptosis. Okay. So regardless of how they're doing it, their EPI-321 is having an effect on reduction of DUX4, reduction of DUX4 target genes, and reduction of cell death, and increase in methylation. Whether or not that matters or not is kind of debatable. And one of the reasons it's kind of debatable is that two things. First off, this is a great paper from Stephen Balin, who was at Johns Hopkins back in 2001. This is back when I was at NIH working, and he was kind of like a friend of me. We were kind of collaborators and competitors. Really great lab on cancer epigenetics at Johns Hopkins. And one of the things that, is very clear is that de novo DNA methylation mediated by DNA methyltransferase 3A and 3B can establish repressive epigenetic states, but it needs to do so during S phase, which is when DNA is being replicated in cycling cells. Okay, that's the first thing. The second thing is that DNA methyltransferases themselves are also transcriptional repressors, right? They bind repressor proteins, and so they can also alter gene activity in the absence of altering DNA methylation. Now, in FSHD, your target tissue and the mass of skeletal muscle that you have, those cells are in G0. They are not replicating. So the idea of establishing epigenetic repression, which is needed to be done in S phase, in cells that never go through S phase, it's kind of a hard one to kind of conceptualize, right? Now, if you're truly infecting myoblasts, as described in the legends for the experiments, those are cycling cells. That's not the cells you have, but those are cycling cells in the lab. So you could be establishing repression in the lab and shutting down DUX4 in a way that you could never do it actually in a person. That's a concern of mine. All right, I had mentioned there was a great paper by a Japanese group. This is from Matsuro Sasaki Honda and Hidetoshi Sakurai's lab, and I actually love this paper. It's a hit and run, and because it shows the proof of concept that if you recruit a transcriptional repressor like CRAB, that's the K that we had in our thing, and DNA methyltransferase to replicating FSHD cells they used to induce pluripotent stem cells, you can methylate and epigenetically change the FSHD region from an FSHD state to a non-FSHD state, ultimately in the muscle cells. But it has to be done while the cells are replicating prior to differentiation. So you could do this conceptually, right? The epigenetic state of the locus can be changed. But again, this is not in a case that's relevant to an FSHD patient, okay? So this is what they're trying to do, right? Go back from an FSHD state to a non-FSHD. And the question is, can this be accomplished with just a little bit of methylation, all right? And in non-replicating cells, I've never seen any data that says that can, but whatever. All right, so now, but most importantly, right, from your standpoint, is increased functionality. What, you want, of course, we want to decrease DUX4 and target genes and apoptosis and increase methylation. That's all well and good. What you really want to see, though, is increased muscle function, right? And so my expectation when they claimed that they did this would have been to have seen FSHD mouse, even xenograft mouse. But instead, they used a 3D muscle structure. And I got to tell you that to me and to colleagues of mine that I ran this by, who are experts in 3D FSHD muscle, honestly, my take-home message is this was scientifically uninterpretable, okay? They claim that this shows the increased functionality, and that's over here. If you add virus containing EPI-321 to 3D assembled FSHD myofibers, you do get an improvement. It is not a dose-dependent improvement, okay? And actually, I'm not sure it's actually an improvement because, again, if you looked at all the apoptosis they were showing and all their cells dying, what's probably going on is the FSHD cells maybe are all just dying off and this is just staying the same. I don't know. I guess that still shows, though, that it's better than non-FSHD. But the bigger concern is that for those who actually make 3D muscle, and they show here that at day 7 is when they actually put the virus in, they say that the problem with 3D muscle cultures is you can't get therapeutics into the cultures. Actually, you have to add the virus when you're assembling the 3D not once it's assembled, because the virus can't penetrate. There's no vasculature. There's no way for the virus. So I'm not sure how they even get an effect other or just a minimal effect. But anyway, the claim is that there's increased function. I hope that's true. This data, to me, doesn't show it, and there's no dose response. I'd be very curious if the DNA methylation changed in these cells and if there was repression. But more than anything, I'd like to see when this is done with healthy muscle and how strong healthy muscle is. Are you really reverting back to a healthy, functional muscle? But still, getting some benefit would be cool. That's fine. You take some benefit, right? Even if it's not all the way back to healthy, you take some benefit. All right, the other way to do it is to look at a muscle xenograft. Now, a xenograft is where you actually grow a human FSHD muscle in an immune-compromised mouse. And you have to do an immune-compromised mouse because otherwise it'll reject the human cells, standard organ rejection, right? Now, there are many ways to do this, but Bob Block at the University of Maryland developed a way that really efficiently makes really great vascularized and innervated FSHD muscle growing in a mouse TA. Okay, you irradiate the hind limb of a mouse. That's going to eliminate all the muscle stem cells from the mouse. Then a few days later, I'm going to hit it with barium chloride or cardiotoxin, which is spitting cobra venom, and that's going to eliminate all the muscle fibers. It just clears out the cells. Then a few days later, and it's important to wait a few days, you add in your human FSHD muscle cells, which are replicating cells from culture. Okay, now you have injected replicating cells into a decellularized compartment of the mouse, which is normally the tibialis anterior muscle. And then what we do is we electrostimulate these mice then over the course of a month to get the muscle to actually be functioning, and it stimulates growth. The cells stop replicating, they fuse, the muscle starts growing, and you still have kind of a very young muscle, but you end up with a functional, contracting human muscle in the mice. And then you treat them. Actually, you can either do an IM injection or a systemic injection, and you can then evaluate these many months later, 4 months, 6 months, however long you want, because the muscle's growing in the mouse. So you can actually do a long-term study in a functional human FSHD muscle, okay, in the absence of an immune system, which is a caveat. All right, so what does EpiCRISPR do? What they do is they do a xenograft model, but they do a very unusual system, okay? They're going to use an immune-compromised mouse. They're going to do the irradiation. They're going to add in cardiotoxin. Then 1 day later, they're going to add in replicating cells. And then 1 day later, while these cells are still likely replicating, they're just growing in a different environment, they're going to add in their virus, their RH74, which is actually going to get into what we've found is that this gets into the muscle, into these cells, much easier than it would if they were actually a fully differentiated muscle. And then after 24 days, they're going to collect and do their assay. So they're essentially infecting muscle cells that are replicating again, and they're assaying, and before that's even actually formed into real muscle, quite honestly. At least they haven't shown that it's muscle. And I'm guessing, it's an educated guess, that they're really assaying grafts. There's huge variability in these grafts. We've done hundreds of these grafts, tremendous variability in grafts, cell line specific variability. But even if you just take in the cell line, you take 10 mice and you make 20 legs, you get 20 different kind grafts. You have to normalize to the graph, and some are very good and some are not so good. That's the whole point of actually going the way we do it. Actually, at the very end, not just a gob of cells that have been grown in a mouse. Right? So the concern here is they're not actually testing the therapeutic on FSHD patient target tissue, not showing repression or methylation in non-replicative cells. And they're really just, it seems like assaying engraftment efficiency. Here's the data that they go through, and the data what to show. Again, we don't know what the doses are, but the low dose does not show any reduction of DUX4 mRNA. The medium dose barely shows any reduction of DUX4 mRNA, and the high dose shows maybe 30% reduction in DUX4 mRNA. Target genes go down about 50% in that high dose. Again, in the low dose, they don't go down at all. DUX4 protein, oddly enough, goes down in the low dose equally well as the high dose, but it doesn't. It actually goes up in the middle dose, so that doesn't make any sense. And then apoptosis goes down in the high dose. So if you just look at the high dose and the untreated, which again is not the right control, by the way, then you can see that they get a decrease in DUX4, decrease in DUX4 protein and target genes, not a tremendously large decrease, and decrease in apoptosis. Now, the proper control should be a non-targeting guide. Remember, we show you that in CRISPR, you have the CRISPR machine and the guide RNA that tells it where to go. The proper control in these experiments is a guide RNA that doesn't target the D4Z4. That way you're expressing the machine, you're expressing everything, you're just not telling it where to go, and that you control for effects due to viral infection and these types of things, and expression of your cargo that are independent of targeting. But again, they didn't do the right control. Again, when I say right control, that's if you were working in my lab. We would do that control, and we always do that control, as you can see in our publications. All right, now this is where the rubber kind of hits the road for a little bit of things. The first time, it hits the road twice. This is the first time that we actually got some comments from people who said, hey, Jones, you told us this was not using a muscle-specific promoter, but hey, they're using CK8E, which is a muscle-specific promoter. You don't know what you're talking about. Now, I love it when people tell me I don't know what I'm talking about. And sometimes they're right. And I got to tell you, I never want to give you bad information. But I think what happened is Carice and I just didn't explain very well how we assess this, because we see this kind of data all the time. Okay, so first off, the point of this study was to show safety in mice, to show that the viral infection and the expression of their EPI-321 cargo had no adverse effects in mice. And I think they actually showed that. The testes were fine, the ovaries are fine. So as a safety study, that's fine. But we looked at it a little bit differently. It also gave you some information about biodistribution of their virus, as well as cargo expression of their virus. Okay, so let's look at that for a second, okay? Now, first off, remember that their in vitro cell culture data showed that if they got 12 viral genomes into a cell, they got 40% reduction of DUX4 and a 30% reduction of apoptosis. Okay, so we're going to do the vector copy number, copies per viral genome. This is the first thing that kind of caught my eye on this dose that they did, which they don't tell us what the dose is. But essentially got one vector genome per muscle nucleus in the TA. I thought, holy cow, that's not very good. Right? And then you looked around and you see, well, they actually got more went to kidney, equal lung, basically a little bit more into kidney, heart a little bit more, liver, a ton goes to liver. And again, this is exactly why you don't want to use AAVrh74. It goes to all of these tissues that are not skeletal muscle, and it really goes to liver, meaning it really infects liver. Okay? So that's the function of AAVrh74. Okay? But the other thing that caught us was the transgene expression down here. Because remember, this is supposed to be muscle-specific expression, and there's reasons you want it muscle-specific. One, you only need it in the muscle. But two, you want muscle-specific expression because concerns with the exposure to the immune system and the immune response, okay? That's one of the reasons. And also potentially because you could do damage to the liver or myocarditis, damage to the heart. Now we say muscle, and you say heart to muscle, and that is true. CK8E is known to express very highly in the heart. And that actually is a problem that everyone in the gene therapy field knows, is that myocarditis, due to high levels of cargo expression in the heart in gene therapy trials for Duchenne, for LGMD, whatever, you pick your disease, high levels of the cargo expression in the heart are not ideal, okay? So we saw this and we said, oh, wow, this is actually not very muscle specific. Now, someone that kind of knows what they're talking about said, no, wait a minute, you got to remember, this is actually transcript number that is normalized to copies of RNA. This is not an expression study per se. You can't just look at this and come up and say, oh, it's expressed everywhere. Now, of course, we do know that. Because we came up with the conclusion, strikingly high expression in liver, lung, testes, and ovaries, actually in kidney as well. So how did we come up with that? So I thought I'd walk you through it, because I do like to take you through some new data. Okay, so one thing to keep in mind, because what they're doing is they're going to look at the transcript copy number, copies of their cargo per nanogram of RNA, okay? And now one thing to keep in mind is that we're going to talk about this question about how they normalize their data to tissue mRNA content for CK8E, which again is a well-characterized muscle-specific promoter that is used in neuromuscular disease gene therapy. This is the promoter that's being used in the FDA-approved DMD drug, Elvidus, right? Okay, so we're going to first start, if you're looking online, on these two graphs here. Now the thing is, and these are going to be based on my estimates looking at their chart on the copies of transcript, okay? And that's over here on the left. Now, one thing to keep in mind is every tissue has a different level of RNA. So now, of course, we know this, so we say, oh, that one is high, that one is low, and so we were ballpark figuring this out in our heads, but this is how we decided we better do the math if we're going to make the claim. So if you just take the average amount of RNA found in the kidney, it's 3,500, in the skeletal muscle, it's 425, in ovary, 1,600, in heart, it's 5,500, right? The amount of the nanograms of RNA per mg tissue is very different for different tissues, okay? So then, liver, is actually quite high. In fact, it's the highest of all, okay? And you take that into account because the amount of cargo per amount of RNA, right? If there's more total RNA in the tissue, that's going to change the percentage of your cargo RNA. So then we just basically multiplied the copies of RNA transcripts that they determined by their digital droplet PCR and the average total of mg RNA and transcript copies per mg of tissue, okay? And that's what we end up with. Copies per nanogram of RNA, copies per mg, transcript copies per mg of tissue is right here, okay? And then we took the estimate of the virus copy number, which was this graph over here on the left, right? And they say, well, on average, in skeletal muscle, they got about 0.9 vector genomes per cell, okay? 1 in the lung, 1.5 in kidney. Again, this is my estimates looking at this graph, okay? 0.2 in the testes, 1.5 in the heart. And then you're going to take the transcript copies per mg of tissue, and you're going to normalize it based on the amount of virus. And this will tell you the expression kind of per viral genome, okay? And that's this over here to the right, kind of a normalized, you just kind of taking into account that some of these tissues like liver has 600 copies of the vector, right? Whereas in the lung, it only has 1 copy. So liver has the highest expression level, but also has the highest number of viruses. You divide the expression level by the number of viruses, and you end up with, actually it turns out to be a much lower number. And that's graphed over here on the right. And what you see is that it's not actually, liver actually kind of goes away, but this is what we had, kind of, again, doing these ballpark figures in our head, we were able to see that, lung was going to be kind of high, ovaries kind of high, testes is kind of high, and heart's kind of high. Okay, heart is a muscle, but the ovary and testes is it's very common for testes to be expressed in these muscle-specific promoters. There's some evolutionary conservation among muscle regulatory elements in testes, and according to Caris. But the striking thing was how high lung was and how high kidney was, okay? So relative to skeletal muscle, you get the numbers down here, and that's this right here. And you can see in the heart, you're getting 58-fold higher expression in the heart than in skeletal muscle. And remember, you don't need it in the heart, okay? You're getting how much in the liver? You're getting 1/50th the level of expression in the liver compared to skeletal muscle, right? So that's okay. The promoter is a little bit active, but not very active in the liver, but it's strikingly active in the lung. And this is what kind of caught us, and in the kidney, and in the ovary. And ovary 17-fold higher in the ovary than in the skeletal muscle. So this is why we said, this doesn't actually look like it's acting quite like CKE, because it's been very well studied. So then we're going to go to questions about EPI-321. Now, EPI-321, you said, weren't you just studying that? No, we were just talking about the actual promoter and where it's active. But now we're going to take into account the AAVrh74 distribution of the cargo. So now if we take that into account, we're going to go to slightly different numbers. We're really going to focus on the transcript copies per mg of tissue. All right, that's actually because, and we're going to ignore the vector copies, because our question is going to be, where does this therapeutic actually express? So that is a combination of the promoter where it's on and where the tissue where the virus delivers it. Okay, and you put those together, and now actually this is where you start to get some additional issues. What you find is that all of a sudden the liver jumps right back up. The liver has got 12 times more expression than skeletal muscle. The heart, 97 times more expression than the skeletal muscle. Lung, 5 times more than skeletal muscle. Kidney, the same expression as skeletal muscle. Ovary, 23.5 times. Right? So again, they showed the ovary and the testes were fine. Again, we're not talking about safety so much, other than, I gotta say, long-term high-level expression in the heart could have some concerns. Myocarditis that is seen in other neuromuscular diseases. This is more going to get to the potential exposure to the immune system and actually potential issues with the liver, as is being seen. Again, the liver issues being seen in gene therapies are due to the high doses of the AAV, but we'll get to that later. Okay? Now, you might be saying, well, Jones, you're just bitching and moaning. It's a muscle-specific promoter that's well documented. Why are you complaining? AAV-RH74 has been used in other neuromuscular diseases, FDA-approved trial. This promoter has been used. What's the problem? And so the problem is, one, it's not actually acting quite like we would expect it to. Okay? And two, there are better things out there. Now, I'm going to show you briefly. What is CK8E? Well, CK8E, because our lab is actually, my research lab has done some work on this. We published this paper, Have a Little Heart or Not, Highly Minimized Skeletal Muscle Regulatory Cassettes with Low or No Activity in the Heart. And I gotta tell you, this stimulated Kate Therapeutics to start investigating this, because the reason the field was very excited about this paper and our work is if you look at CK8E and CK8, right, which are typically the promoters that are used in neuromuscular disease gene therapy, right? And they're similar. The only difference is CK8 is 3 copies of CK8E, right? The CK8E is a single copy, so CK8 is 3 times better, but it's a little bit larger. If you go over here to the right, you see when we put CK8 into a mouse, you get very high expressions in the heart, right? And it doesn't matter how you deliver, you get very high expression in the heart. Everybody in the field, kind of like the dirty little secret, knows that that's not good. You need maybe some expression in the heart for Duchenne, but a lot of these diseases don't need expression in the heart or just need a very little in the heart. And what Carice did when she made Have a Little Heart and No Heart, she made skeletal muscle specific promoters that truly are skeletal muscle specific, and we're going to show you that, but also they don't express very much in the heart, which is cardiac muscle. Okay? She took out the car domain, right? This card box right here. And this is the way we look at it. So this is the TA, the EDL, the soleus, the GA, the quad, abs, and heart. And what you see is CK8, which again, CK8 is 3 times higher expression compared to CK8E. So that just so you're aware. But you can see while it is very highly expressed in skeletal muscle, it's incredibly highly expressed in the heart. We took an FSHD specific approach, and where you don't need to have this expression. One, it's safer for anybody to have lower expression in the heart, and in FSHD, you don't need it in the heart. And so now if you go and let's look at the expression that's over here on the right, liver, kidney, brain, testes, heart. The thing that's kind of striking that you might catch is that kidney and liver, which have a lot of viral transduction, have almost no expression of CK8. Okay, this is 3 times higher than you'd see with the others. It's almost essentially undetectable, which is not what they actually saw with their CK8E, which is actually a much weaker promoter than the one we're assaying here. If you look at the full tissue, we use a red reporter, M-Cherry reporter, and so the cargo for the no heart, the have a little heart, and the CK8 is glowing red, and you can see the gastroc, the quad, abs, down here CK8, see the heart glowing like crazy, right? Like a hot coal, TA. And CK8, right? The lung, undetectable. Liver, undetectable. Ovary, undetectable. It doesn't really appear to be on, right? That's not what they saw, right? That was why we were concerned. I said, wait, is this really CK8E? Now, maybe it is, or we don't know. They say it is, and that's fine. But that's why we were concerned. So people who kind of said, hey, why are you criticizing? They've said, I don't care what you say you're using. I don't care what you actually do use. I just look at the data. And the data suggested it's not functioning like we would expect CK80 to function in the mouse. Okay, and that's from our own experience, okay, as well as the published literature. And we went back and looked at the original papers by Jeff Chamberlain and Stephen Hauschka, and they didn't see it either. So there's something wonky going on. Again, that's our interpretation of their data. So for those of you who commented on that's why we made that indication. But what this also shows is you can get high levels of skeletal muscle expression here, have a little heart. It's very specific, soleus, EDL, TA, gastra, quad, and very low in the heart. This is going to be much better for a gene therapy recipient, regardless of what disease, DMD, LGMD, FSHD, this is going to be much better. And this is actually why so many companies are excited about Carissa's Have a Little Heart promoter, because not only is it small, but it's actually much better than CK80, frankly, or CK8. Anyway, so the CK8 promoter used in the literature, in DMD clinical trials, EpiCRISPR's preclinical data in our hands has exceptionally high transgene expression in the heart. Everybody knows that that's true. So even if you ignore everything I said about lung and kidney and testes and ovary, and say, well, that wasn't an expression study anyway, so you're picking at them for something that was a safety study, that's fine. You can ignore everything I just said. There's still a concern to me about the incredibly high level in the heart. And many companies in the field have the same concern. That's just if you just make the assumption CK8E as a promoter is functioning as it's supposed to. Okay, so overall, my concerns about EPI-321 from this first part, tissue expression pattern in mice is highly unusual for the CK80 expression in mice, right? If you take into account the RNA levels that I just showed you, the viral genomes, it's not actually recapitulating what's published for CK80 from the Hauschka and Chamberlain labs who developed the cassettes, and Hauschka's lab is where Carisse came from. That's why we pointed it out. Expression in the lung, testes, and ovary is still very high. And again, we're not saying it's not safe. We have other reasons we think this is going to affect functionality and durability of the therapeutic. Some transgene expression in the liver may be good. So, we're not to hammer transgene expression in the liver. You want potentially a little bit of it, because a little bit of liver transgene expression can tolerize the immune system so that you don't get an immune response. But high levels in the liver is not good. The thing that was really striking, though, is that if you look in the literature, the difference between liver expression and skeletal muscle expression is 500, 600-fold. And you're only seeing like 30, 35-fold. So either it's not high enough in the skeletal muscle, or it's too high in the liver, or it's kind of both meeting in the middle. Anyway, it told us, it was something that just struck me as like, wow, that's kind of wonky. And again, maybe it doesn't matter. That's fine. We're not saying it's not safe. Level of transgene expression in the heart is very high. But again, this is typical of the CK80 promoter, and it's typical of actually exactly what they showed. Regardless of design or intent, it's very high, and that gives the potential for myocarditis. Transgene expression is not needed in the FSHD heart, and better promoter options are available, as I just showed you. Overall levels of transgene expression in the liver as a whole is high, right? The whole liver itself is actually kind of high. And again, I showed you what we consider the liver should look like in the Have a Little Heart paper that Carisse did. Okay, so it's high because of the AAVrh74 targeting, which is one of the many reasons you don't want to use AAVrh74, in my opinion, when there's other better viruses that are available. CK8E is in the microdystrophin DMD gene therapy, right? And experts were, again, Chamberlain, Hauschka, these guys, they're world experts in this stuff, and they use this. But again, in DMD, you need expression in the cardiac muscle. We only need to shut down DUX4 and differentiate skeletal muscle. Okay, so regardless of what promoter sequence was used or intended, the data, in my opinion, speaks for itself. In the context of EPI-321, meaning the context of AAVrh74 plus their version of CK8E, in the context of the guide RNA upstream of it, in a packaged AAV genome that is going to be then released into the nucleus, in that context, combining the cells and everything there is, it does not appear to be very muscle specific to us. Okay, is that a concern? Maybe it could be. Is it a safety concern? Not likely, but it is a concern potentially for efficacy. All right, so now we're going to move down. Again, I'm running behind. We're going to see if we can speed it up a little bit. Here we get a safety study in non-human primates. They're going to do groups of 4 animals, 2 males and 2 females, and they're going to do essentially untreated, vehicle treated, EPI-321 alone, virus infection. And they're going to do it with prednisone essentially, or a triple immune suppression regimen of prednisone, sirolimus, or rituximab. Okay, now the thing to remember is they actually make a note up here that high dose AAV is required for effective muscle delivery, right? That is actually a fact, right? Everybody knows that, right? You need high dose AAV for effective muscle delivery, okay? And so they're going to use, we'll get to their dose in a second. What now they're going to do here is they're going to have a triple immune suppression. When they use rituximab, that's going to eliminate B cells, so the body can't produce antibodies. And essentially through the course of this test, in these triple immune suppression monkeys, they're not going to be able to produce antibodies, they're not going to B cell response. The sirolimus is rapamycin, mTOR inhibitor, and that's going to prevent proliferation of B and T cells. So essentially you're not going to get a T cell response. And then the corticosteroid is going to be anti-inflammatory, and so you're not going to get an inflammatory response. Okay? When they did this, they found that EPI-321 is safe and well tolerated in all cases with the non-human primate. So that's fantastic. That's a 39-day study. Okay. And it was generally safe with the prednisone as well as the triple immune suppression. So that's fine. Again, we're not saying this isn't safe. But there was some interesting data they found. So first off, they're going to use a dose of 1.2 times e to the 14th vector genomes per kg. Okay. And this is going to be 6 times the initial clinical dose, which is 2 times 10 to the 13th vector genomes per kg. It's 6 times that. This dose level, they found no adverse effects. Okay, and that's great. Again, we're not saying this isn't safe, right? But we are going to look at some of the data that they have. So first off, we want to focus on the dose. And now the clinical trial is going to have prednisone. So we're going to focus on the blue one here if you're looking online. And it's either going to be 1/6 or 1/3 of the data they're actually showing here, okay? So the data they're showing here is that transgene copy per nanogram of DNA looks to me like it's around 120, okay? 120, okay? Why is that important? Well, you know that we like to do math. I like to do math, right? And I can say with a transgene copy number of 120, per nanogram of DNA. We know in humans, and so we're going to say human is similar to primate. In humans, 152 cells for 1 nanogram of genomic DNA. So at their dose of 1.2 times 10 to the 14th, right, using prednisone, they're getting about 120 copies of their transgene for 152 cells. So they have less than 1 copy per cell. Now remember, in the preclinical data, 12 copies per cell gave you a 40% reduction. Here they're going to average less than 1 copy per cell. Now you might think, oh, you just need to get 1 copy in. That's just not how it works in gene therapy. You need to get more than 1 copy. This is on average. So a couple of cells might get 5 or 6 copies that are near the blood vessels. Other cells might not get any, right? It's just kind of on average they're getting less than 1. Okay, so it's not very good. And this is 6 times higher though than what they're going to put into a patient. Right, so if you just kind of arbitrarily say, okay, well, let's divide it by 6, they're going to have less than roughly 20 copies per 152 cells. So essentially 1 viral genome per 8 myonuclei, right? Or 1 viral genome for 4 at the high dose. Right? This is actually an incredibly low dose. And if you looked at the longer podcast, you'll see that in the dog studies for Duchenne, this essentially led to undetectable levels of the cargo and virtually no muscle fibers that were showing positive. Less than 5% of muscle fibers are positive around about these doses. Okay. Now in the clinical trial, they're going to do, again, as I mentioned, 2 times 10 to the 13th and 4 times 10 to the 13th. Okay. So this is very, very low dose, likely very safe. Okay. But the question is this enough to get enough activity into enough cells to flip enough of the epigenetics to go from FSHD to non-FSHD? And again, 12 copies per cell, right? Because again, not every copy is going to be active. Not every copy actually gets in the nucleus. Not every copy, and you do need to, these have to crank out and make the protein. Okay, not every one good. So in an idealized situation in the lab, where likely every cell was infected with 12 copies, they still only got a 40% reduction in DUX4, right? This is way, way lower, okay? Now let's focus on durability, right? And that's why I've been talking about this muscle-specific issue for a while, right? The data over here on the right is actually looking at expression of their DCAS9 cargo, okay? And if we focus again on the blue one here, which is the prednisone, right? And again, keeping in mind, this is 6 times higher or 3 times higher than the clinical doses, right? What they show is that prednisone versus no immune suppression is about the same. You get a level of about 50 copies per nanogram, right? In triple immune suppression, it's up over 300. So 6 times more if you triple immune suppress, okay? Meaning if you eliminate the B cell response and you eliminate the T cell response, you get many, many more cells producing the cargo. So why would that be? Well, there's this paper we've talked about, the Cas9 specific immune responses compromise local and systemic AAV CRISPR therapy in multiple dystrophic canine models, okay? What this is by Daosheng Duan, from Charles Gersbach, some really good people are on here. What they show is that treatment with AAV CRISPR therapy in affected dog induces muscle inflammation and Cas9 specific humoral and cytotoxic T cell response that are not prevented by muscle specific promoter and transient prednisone, right? So the reason you need the muscle specific promoter is you're trying to do everything you can to prevent immune presentation and developing an immune response that is going to then go affect the muscle cells. But what they're saying here is even if you have the best case scenario with a really good muscle specific promoter and muscle specific expression, which they don't, you still stimulate an immune response when you're delivering Cas9 to muscle cells. They say our results suggest Cas9 immunity may represent a critical barrier for AAV CRISPR gene therapy in large mammals. You're a large mammal. Without going through the data, I'm just going to you can go back and look at it online. But if you actually look at the T cell response at 3 weeks, you're starting this brown staining here, you're starting to get an immune response to the cells expressing the Cas9, okay? This is in dogs. If you look at 6 weeks, you're really getting a strong response, and by 12 weeks, you're getting a tremendous response, and you're expressing granzyme B, which is a protease that's associated with cell killing, right? T cells and natural killer cells are actively killing cells that are expressing Cas9 at 6 weeks and 12 weeks. If you go through and you do the experiment and you look, and I'm going to focus on these data right here that I circled in red and say, what are the viral genomes? Well, at 3 weeks after infection, they have a lot of viral genomes, okay? In the muscle, they have over 60. But at 6 weeks post-injection, actually you're starting to clear the virally infected cells. That's at 6 weeks, right? Okay, if you look at that's at viral genomes, and you look at transcripts, same thing. The transcript is starting to go way down. You're starting to express less cells. If you look at 12 weeks, it goes to almost zero. You have almost zero infected cells remaining, and you have almost zero expression of the cargo. Okay, by 12 weeks, you've almost completely eliminated the cells that have been fixed by the Cas. It's not that the Cas is gone. The cells expressing the Cas are wiped out by the immune system at 6 weeks and 12 weeks. The data that EPI-CRISPR did is 39 days. They don't even go out to 6 weeks. And then if you go and you look for antibodies, what you see is after injection, almost immediately upon AAV injection, you start developing anti-Cas antibodies, okay? So if you look at the EPI-CRISPR data, which was not even at 6 weeks, right? 39 days, 42 days would be 6 weeks. You're seeing the same thing, right? What you're actually seeing is that if you triple block the immune response, you get much better expression of the cargo and you maintain the virally infected cells much better. What would this, do they actually look? Because this suggests to me, my interpretation of this data is you're developing immunity against DCAS, Onyx, DNMT expressing cells, leading to their elimination. That's what it's suggesting to me, which means that even if you ignore everything that I've already told you about, and this everything is going to work, the cells that get repressed and silenced are going to be eliminated. That's what the data suggests to me. And you can figure that out before going to trial. So the take-home message from this data in dogs on Cas9, they used different Cas9. And again, they didn't test Cas Onyx. They just tested their own other natural cases. But they said that importantly, we showed that strategy commonly used to minimize cellular immune responses, tissue-specific promoter. That's why we're hammering it. That's the immune response. That's why you only want it in skeletal muscle. The fewest, you only want it in the cells that you need it to avoid the immune response. But even if you have a great tissue-specific promoter, even if you have prednisone, these are insufficient to bypass Cas9 immunity. Comprehensive approaches, including but not limited to immune-attenuated, immune-privileged Cas9 expression cassette, non-viral vector-mediated transient Cas9 expression, immune tolerance induction, a little bit in the liver is probably good, and more potent immune expression may be needed, right? This is one of the reasons, again, why these myotropic vectors are being developed and things. Right? A lot in gene therapy, a lot of focus is on the immune response to the virus and immune response to the cargo. Okay, so the conclusion of the EpiCRISPR preclinical data is right here, that a high dose 1.2 times e to the 14th is safe and well tolerated. They didn't find anything unusual. Immunosuppression with prednisone was beneficial, and then they're ready to go to trial. Okay, so let's put it all together. Okay. Concern 1, the doses may be safe, but I'm concerned that they will have little, if any, chance of being efficacious. Okay, I don't know what evidence was used on deciding this dose in clinical. I didn't see anything. Now, again, I'm not aware of all the data, but I haven't seen anything that suggests these dosings will be effective. I didn't see any true efficacy experiments in vivo. Biodistribution and durability for the clinical trial in NHPs was not performed that I'm aware of, and the efficacy for the clinical trial, that, I mean, the xenograft dosing, I mean, we don't know what the doses were, but it was not very impressive data. Okay, I suspect that the very high doses that they needed, could they do relevant low doses? Because their low dose and the mid dose didn't have any benefit. Concern 2, ignoring all of that I just told you, pretend that everything is going to work fabulously. Pretend the dose is great. Pretend it works very well. Pretend it shuts down DUX4 by 99%. The concern is, will the benefit, if any, be durable due to clearance of cells expressing DCAS9? Will the DCAS Onyx protein be durable? What happens to cells expressing this? The expression profile beyond skeletal muscle presents additional immune concerns, right? It's great that they use CK8E, but their expression profiling suggests that they're getting some other tissue expression that may not be very good, especially in the liver and the kidney and other places. Biodistribution supports that AAV-infected cells are being cleared. That's what the data suggests to me. Okay. Did they investigate if there was an immune response or clearance? Basically at day 39 is a little early, but they could have looked. Did they look? What's the percentage of cells that are positive for their Cas Onyx at day 39? Did they get antibodies to the Cas Onyx at that time? They should have been able to detect it. Did they look? Did they know? Or did they not want to know? Let me tell you, I've consulted for other companies. I don't have any connection with them, but I've consulted for companies that don't want to know the answer. Once they've decided to go to clinical trial, they don't want to know the answers to any question that's going to say, yeah, maybe you should go to clinic. Okay, are there antibodies? All right. So we're gonna take a brief break. We're gonna give you some rockabilly, and then I'm gonna come back. We only got a little bit left, but this is gonna be the Vidalias from Bubba-Palooza 1995, Star Bar, Atlanta, Georgia, faking it.
I was quite impressed. You never criticized my car or the way I dressed, and you never seemed to care if I had money in the bank. And when I took you out, you just said thanks. And as I got to know you better, I was quite intrigued. You never seemed to blame the world for all your unmet needs. You see through. Well, I was kind of disillusioned when it finally hit. You really ain't so great, you're just faking it. Has me quite dismayed. You never miss a chance to contradict the thing I say. You bend my ear for hours on end, complaining how you feel, and you tell your therapist that I'm a heel. Well, something should have told me you were too good to be true, but you put on a happy face, but I could not see through. Well, I was kind of disillusioned when it finally hit. You really ain't so great, you're just faking it. You really ain't so great, you're just faking it.
All right, so back. I hope you like that. I love my rockabilly. Good stuff. Starbar was a great place to go during graduate school in Atlanta. Bubba Palooza, some of my favorite bands got to play there. Redneck Grease Deluxe, Southern Culture on the Skids, some good stuff. Anyway, we're going to talk about this EPI-321 trial. Make no mistake, this is an FDA-approved clinical trial. It meets all federal regulations for safety at this stage, and the FDA accepted the preclinical data package. So I don't know if they share my concerns or not, but you keep in mind, these are my concerns, Carice's concerns, and we're not we're not the FDA, we're not a regulatory body. We are just independent people looking at this as scientists, evaluating it like we would a paper or a grant, and these are our concerns, okay? And again, nothing we have said or implied suggests there's any issue with safety. All right, so since I'm talking concerns, one of my concerns is, you might know there's something called the FSHD Clinical Trial Research Network. These are sites all across the United States, all across the world. There's sites in Australia, there's sites, I don't think there's one in New Zealand, but there are sites across Europe and such. These have the world's best FSHD neurologists, scientists that are used to evaluating FSHD efficacy and other clinical trials, tremendous expertise in biomarkers, MRI, functional outcome measures. The whole entire system is set up so that you can run a fantastic FSHD clinical trial and truly evaluate efficacy of your therapeutic. And EpiCRISPR is not using that resource at all anywhere. They've chosen not to. They don't have to, and they're not gonna. I'd like to know why, but they don't have to say why. But you might want to know why too, because that's shocking to me. Okay, study overview. The goal of this clinical trial is to learn how safe and tolerable EPI-321 is and whether there may be early signs it is working in male or female participants with FSHD type 1. The main questions it aims to answer are, how safe is EPI-321 and how well can people handle it over time? How does EPI-321 interact with its target and does it show early signs of working? Okay, those are actually great questions. I think the question is, does the experimental design answer those questions? Okay, that's my question. Okay, description, it's a product comprising recombinant AAV RH74, we know that. It has been shown to transduce human skeletal muscle efficiently in the clinical experience. That is true, but it also transduces liver efficiently and transduces all these other tissues as well, which is why you need to have such an incredibly high dose. The DCAS Onyx is fused to an epigenetic modulator that is going to bind D4Z4 and methylate the CPG. They're doubling down on methylation. I really hope they have the crab repression domain in there, and I really hope they're using the repressive activity of the DNA methyltransferases, because I don't think the methylation, 8.5% of methylation alone on non-replicating cells is going to have any effect. That's my opinion. But they're going to look at methylation on chromosome 4 and repress DUX4, ameliorating the downstream pathology. As is under a muscle-specific promoter, it is expected to preferentially and actively be expressed in muscle tissue following IV. It is expected to. The data we saw says, it'll be expressed as well in skeletal muscle as it is going to be in kidney. It's going to be expressed better in lung, and it's really going to be expressed in know how they say muscle tissue? Cardiac, heart's a muscle. It's going to be expressed gangbusters in the heart. Now, granted, a human profile is a little bit different than a mouse, but the mouse data is concerning. All right, so here's, we already covered this. They're going to be doing 2 times 10 to the 13th vector genomes per kg and 4 times 10 to the 13th vector genomes per kg. Their main outcome measure is going to be adverse events. And honestly, I think they're going to meet this. I think that they are going to find that they have very little, if any, adverse events. It's a very low dose of a therapeutic that's likely going to be cleared. Their secondary outcome measures, they're going to look at vector copy number, just like they did in the non-human primates. They're going to look at cargo transcriptional activity, just like they did in their non-human primates. Then they're going to look at DUX4 expression, which they can't do in the non-human primates, and methylation status, again, which they can't do in the non-human primates. Now, my issue with this is these first 2 can be done and should be done in non-human primates, in pigs, in dogs. Don't do it in dogs. Non-human primates or pigs. Now, the reason one might not want to do this, I view this as an academic and as a business person as a go, no-go experiment. If your DCAS Onyx is getting cleared by the immune system, your program's done. You don't go to clinic. There's not going to be any efficacy, right? If you don't get any cargo transcriptional activity because it's being cleared, it doesn't matter your dose is too low, dose is too high, whatever. If you can't detect it, it doesn't matter. You don't go to clinic. You can figure all of this out in the non-human primate. You can figure this out in a pig, and you can figure this out before you go and stick it in a person. The problem is, if the answer is it's getting cleared and the fixed cells are gone, you don't go to clinic. I think that's important. Because why would go to clinic with something that's got no chance of working? But alternatively, you can go to clinic and you can say, now I'm a clinical stage company, and just put your fingers in your ears, close your eyes, and hope that it works. Do the experiment in people. I would want to know. I guarantee you, I will know before I go to a clinical trial if I were doing this. So what are my comments, questions, and concerns? Conceptually, for FSHD1, I would expect that you need a viral dose at least in the range that's used in DMD, to reach enough cells and fix enough myonuclei to shut down DUX4 expression in enough cells. This is a strikingly low clinical trial dose based on their own NHP biodistribution studies, right? Their 1.2 times 10 to the 14th showed less than 1 viral genome per myonucleus. And they're going to go 6-fold and lower, right? Their in vitro data showed that 12 viral genomes per cell only showed a 40% reduction. They're going to be 50 times lower than this. They expect it to work? Really? Now, I got to tell you that, when again, we're not talking about safety. The safety data, there's there, but there's no efficacy data. I don't see anything in any of this that suggests these are going to be efficacious doses. In fact, data in dogs and humans support that these are not going to be effective doses. Not going to get any benefit from this. That's what the data predicts, and that's what I predict. Durability studies on, again, granted, different Cas9s in dogs find an adaptive cellular immune response to expression and ultimately elimination of cells expressing Cas9 cargo, 6 to 12 weeks. Is there any data at all to support durability of expression? Because the data that they show is pointing towards lack of durability. That's what the triple immune suppression data that shows such a great benefit suggests. Okay, to me. That's what it suggests to me. Okay. Triple immune suppression suggests there's immune clearance of these expressions. That's a concern. Now, maybe they have data that says that's not a concern. I sure hope that they checked the antibody response. I sure hope that they looked at the histology and said, hey, Cells are expressing great. Granted, it should have gone out to 12 weeks. What six more weeks, really? Would it have made a difference? Yes, it would have made a difference scientifically. Amber has had an interview. She says engaging with the FDA for a new drug modality requires bold evidence-based approach. I absolutely agree with Amber. She couldn't be more right. She advises biotech, follow the data, follow the science, and advocate for scientifically sound results. I absolutely agree with you, Amber. But you know what? I bet she's a fantastic CEO. I've actually met her a few times. Very nice. I don't have issues with her. I think she's doing a great job. I just wish scientifically the company had done a better job, gotten a better viral capsid, made sure their promoter's doing exactly what it's supposed to be doing, checked everything out in non-dividing, non-replicating cells, and really figured it out. But it doesn't matter. They are in people. They are a clinical stage company. They have done the first in-human investigational gene therapy trial for FSHD epigenetic modification, right? This is back in August, so it's been about 6 weeks. I don't know how many people they've injected, but they are in people, right? And we will find out in 2026 if any of my concerns are valid. I hope that I hope that my concerns are not valid. I genuinely do. Honestly, anybody who knows us knows that we help the competition, we make tools and give them away. We just want effective therapies for you. I hope this works. I hope my concerns are just that of a rambling academic, but I got to tell you, boy, I just don't see how this is going to provide benefit under this dose in this viral capsid. I really wish they'd change it up. So what am I going to say? And this is my quote. I say, our research lab all showed almost 10 years ago that CRISPR inhibition is a powerful gene therapy technology for repressing FSHD pathogenic gene expression, and FSHD is ideally suited for this type of gene therapy approach. However, translating the clinic in a meaningful way for patients takes time, and the devil truly is in the details. As we have seen with other neuromuscular disease trials and extensively in preclinical work, there are narrow margins between safety, efficacy, and lack of benefit, long-term or short-term. Any therapy needs to be both safe and efficacious. Fortunately, the gene therapy space is moving quickly, with many companies learning from past failures and limited successes, such that the industry is now equipped with new and better viral delivery platforms and muscle gene regulatory cassettes for safer and more effective NMD gene therapy. In fact, many are now available. In addition, there are now many tools available to FSHD researchers and industry for those who wish to utilize them to go to clinic with the best possible predictive data for safety, dosing, and efficacy. Unfortunately, in my opinion, it appears that EpiCRISPR biotechnology has chosen to forego many of these advances and tools in favor of older, less effective technology. In addition, I find the preclinical data lacking in many respects. As being witnessed with recent DMD gene therapies, in the long run, patients will benefit most from going forward with the gene therapies that were developed taking advantage of the best available science and tools and technology with the most rigorous preclinical data. While time is muscle, wasted time is wasted muscle. The words that most aptly come to mind are the mantra, if you want to get there fast, go the long way. Overall, in my opinion, I find the preclinical data to be weak and unconvincing for going to trial, and the clinical trial dosing unlikely to provide any benefit, even if the prior concerns were addressed. However, I do wish the trial participants the best, and I continue to look forward to a future with safe and effective gene therapies for FSHD. That's my quote, and I'll leave it at that. Keep in mind, as people are commenting about this, I told you my conflicts of interest. I think it should be made clear that EpiCRISPR partners is a corporate partner with the FSHD Society and Solve FSHD. Okay, I don't know what that gets them. I don't know what that means, but just be clear. Understand. You can say we're competing with them. Well, I could say our critics are partnering with them. In the end, I hope everybody just wants safe and effective gene therapies to get to FSHD patients, right? I understand some people are in the money-making business. My friend Eric told me you can still make a lot of money helping people too. Let's just hope that that's what's going to happen. However, there are some good foundations that I'm still very much supportive on your side. FSHD Global, Friends of FSH Research, FSHD Canada Foundation, Brave in Brazil, and Me FSH in France, Chris Carino Foundation. I know there's others all around the world. There's a good one going, getting up in Spain. You got the one down in Chile. My friends in Vietnam are getting one together. There are a lot of good foundations that are on your side. But always be aware, as we just saw, some foundations are also on the side of the company. Okay, everybody should be on your side. All right. Anyway, feel free to reach out to me with any questions or concerns. I always like to hear how stupid I am. It's always fun. I really, actually, more than anything, I want to make sure we get it right. It's about getting everything right. All right, let's have a healthy debate about therapeutics in FSHD space. See y'all later.
Thank you for listening to the MyFSHD podcast, where we share with you the latest news and information on FSHD as we strive for a cure. You have a question for Peter? He'd love to hear from you. You can reach him at peterjones@med.unr.edu. Thank you for listening, and join us again next time for more news and information.